All right, another exciting addition. I think that's like the term I use or the phrase I use every time we start one of these. But today we're going to talk about the history of regulation in the US, and I'm going to explain to you how biotech products are regulated. So that includes a brief summary of the roles of three federal agencies. So the Food and Drug Administration, also known as the FDA, the U.S. Department of Agriculture, which is the USDA, and the Environmental Protection Agency, EPA. Regulation has always been seen as an important responsibility of government, and the regulation of food in the United States dates from early colonial times. Regulation of drugs did not begin until 1848, and it began with the inspection of imported drugs. Government is well positioned to ensure safety due to its resources, which include laboratories for testing, their ability to collect expert opinion and data from the entire country, and the fact that they can represent the interests of both stakeholders, the consumer, and industry members. The slides that we're going to go through are going to show some of the important milestones in the history of the regulation of drugs in the United States. So in 1901, two medical tragedies occurred that spurred the passage of the Biologics Control Act in order to prevent similar tragedies from occurring. In St. Louis, 13 children died of tetanus after receiving a contaminated antitoxin. And then the second event occurred in Camden, New Jersey. 9 children died of tetanus after receiving a contaminated smallpox vaccine. At the time, there were no central or uniform controls in place to ensure medicine potency, which means the strength and purity, nor were there inspections for testing the final product. So here's an ad for Miss Winslow's soothing syrup. This advertised that it was a cure for teething and colicky babies. The syrup actually contained morphine, and many infants died after being given too much syrup. So in 1912, the Shirley Amendment was created to prohibit false therapeutic claims after these infants died. So between 1906 and 1938, a number of legal proceedings involved unsafe drugs. This demonstrated that existing drug safety laws were not sufficient, and additional laws and amendments were passed, including the Insulin Amendment. This amendment required the FDA to test and certify purity and potency of this life-saving drug for diabetes. In the same year that the Insulin Amendment was passed, tainted medicine tablets resulted in nearly 300 deaths and injuries. In response, the FDA strengthened its manufacturing and quality controls, leading to what would later be called good manufacturing practices.
In 1957, thalidomide, a new wonder drug, was introduced in Europe. It provided relief to grateful, expectant mothers suffering from morning sickness. Sadly, the medication was causing many thousands of children across the globe to be born with severe deformities. We do not know for certain how thalidomide caused birth defects. It may have shut down blood vessels in growing babies, causing abnormal development of organs. The tragedy of thalidomide babies was largely prevented in the United States by a new medical reviewer at the FDA named Dr. Francis Oldham Kelsey. In 1960, Dr. Kelsey, a pharmacologist and physician, had only been at the FDA for a month when the application to sell thalidomide under the brand name Kevadon came across her desk. She felt the drug company's evidence was lacking and insisted on having the scientific proof that thalidomide was safe before the application could be approved. Over the next year, the drug company often complained to her superiors, yet the FDA stood by her decision. Toward the end of 1961, researchers in Germany and Australia were beginning to link thalidomide to the sharp rise in certain types of birth defects, and countries feverishly pulled it from the marketplace. Dr. Francis Oldham Kelsey was hailed as an American hero. A grateful President John F. Kennedy presented her with the Distinguished Civilian Service Medal. Because thalidomide was sold in dozens of countries, we will never know the number of babies that were never born because of the drug. What we do know is that countless others were saved by an everyday hero. The worldwide thalidomide tragedy did lead to some positive reforms in the United States. The Kefauver-Harris Amendment to the 1938 Food, Drug, and Cosmetic Act was a direct response to the thalidomide tragedy. The 1962 legislation required that all new drug applications demonstrate substantial evidence of the drug's effectiveness. In other words, the drug had to show that it would do what it was advertised to do, in addition to being safe. The Drug Efficacy and Safety Initiative followed. It mandated that drugs approved between 1938 and 1962 be reviewed to establish that they worked. Drugs that were shown not to work could be removed from the market. In July of 1998, under the absolute strictest safety standards, thalidomide was approved by the FDA to treat a skin problem caused by leprosy. In May 2006, it was approved for the treatment of a type of cancer called multiple myeloma. It's currently one of the most highly regulated drugs in American history.
Because of the history of the sale of contaminated or dangerous drugs, researchers in the new field of biotech wondered if GM products could harm humans or the environment. There was no uniform or established way to evaluate safety, so in 1974, researchers imposed a voluntary moratorium, which means that they stopped work until there could be a full debate of the issues. In 1976, the NIH, so the National Institutes of Health, Recombinant DNA Advisory Committee was established to review experiments and issue guidelines. Until 1984, the NIH Recombinant DNA Advisory Committee was a federal body that reviewed and monitored drug or DNA research. In 1984, there were many questions about the safety of biotech research and its products, much like today, and a legal challenge forced the government to propose more specific policies to guide their review. In 1986, the year your favorite teacher was born, the Office of Science and Technology Policy issued the Coordinated Framework. It described the regulatory policy for ensuring the safety of biotechnology research and products. Under the Coordinated Framework, 3 federal agencies share primary responsibility for regulating biotechnology: the FDA, the USDA, and the Animal and Plant Health Inspection Service, and the EPA. The FDA is the leading regulatory agency and is responsible for food, feed, food additives, veterinary drugs, human drugs, and medical devices. The USDA is responsible for plant pests, plants, and veterinary biologics. And the EPA is responsible for microbial plant pesticides, new uses of existing pesticides, and novel microorganisms. So new microorganisms or things that have been genetically modified. Whereas the Coordinated Framework for Regulation of Biotech sets policies for product oversight, biotechnology companies are responsible for meeting those regulations. Some consumers see this aspect of the coordinated framework as lacking in safety because it appears that compliance is voluntary. But since the FDA has the authority to remove any product from the market, biotechnology companies do not view compliance as voluntary. The biotech products you have made in this course, such as the cheese and the root beer, and when we do genetically modify our bacteria, will be examples of some of the products that these committees or organizations would oversee. The FDA has broad authority to initiate regulatory action if a product fails to meet safety standards. In 2000, the FDA issued guidelines that require the labeling of GM foods if the food meets the following criteria. It has a significantly different nutritional property. It includes an allergen that consumers would not expect to be present, for example, peanut protein or soybean product, or it contains a toxicant beyond acceptable limits. So not necessarily that they have to put a GM sticker on it or GMO sticker on it, but that they have to include that in its ingredients list. In 2001, the FDA proposed voluntary guidelines for labeling food that does or does not contain GM ingredients, so voluntary to the company. In response to the use of new genetic techniques that involves animals, the Federal Food, Drug, and Cosmetic Act mandated regulation of GM animals by the FDA in 2000. So let me say that one more time. So in response to the use of new genetic techniques that involve animals, the Federal Food, Drug, and Cosmetic Act mandated regulation of GM animals by the FDA in 2009. So the FDA is now responsible for any genetic techniques that would involve animals.
Safe and effective drugs are essential to caring for your patients. So here are 5 things to know about how FDA approves a drug for use. It starts when a drug is investigated and screened. Generally, new drugs are tested on animals before people. Additionally, in some cases, multiple species are used to gather information on the safety of the compound being investigated. The drug sponsor submits an investigational new drug application, an IND, to the FDA based on the results of the initial testing. The application is required to include the drug's composition as well as the details of its manufacturing. The IND presents a plan for testing the drug on humans. The FDA reviews the plan to determine if the proposed clinical trials will put human subjects at unreasonable and significant risk of illness or injury. Generally, there are 3 phases to a clinical trial in humans. Phase 1 emphasizes safety and typically includes 20 to 80 healthy volunteers. Phase 2 emphasizes effectiveness and typically includes hundreds of patients with a certain disease or condition. Phase 3 typically includes thousands of patients and is used to gather more information about the safety and effectiveness in different populations and different doses. After the clinical trials are complete, the FDA looks carefully at all the animal and human testing data, reviews the drug labeling information, and inspects the facilities where the drugs will be manufactured to determine if the new drug is safe and effective. Because it's not possible to predict all of a drug's effects during clinical trials, monitoring safety issues after drugs get on the market is critical. The role of FDA's post-marketing safety system is to detect adverse events and take regulatory action when needed. That's 5 things you need to know about the drug approval process.
In 2000, the regulatory process was updated to consider the possibility that GM plants could be harmful to other plants. The amended Plant Protection Act was passed, and it mandated GM plant testing by the Animal and Plant Health Inspection Service, which is part of the USDA. This act defined GM varieties as potential plant pests. GM-based varieties that harm other plants after field testing will continue to be regulated, but those varieties found that do not are deregulated and are therefore available for cultivation and sale. The USDA also certifies organic food labeling, and in 2001, the department decreed that foods labeled organic must not contain GM products. The EPA has a responsibility for regulating pesticides, which are substances that are used to kill unwanted insects that harm plants. One kind of gene added to GM crops can allow the plant to make its own pesticide. In 2001, in response to this new aspect of GM crops, the EPA began regulating GM plants with pest-resistant traits to make sure that pesticides the plant makes are safe for human and animal consumption and do not pose unreasonable risks of harm to human health or the environment. Tests that ensure that the plants are safe to eat are as follows. So they have to determine the safe level of human and animal consumption of the pesticide, examine the distribution and connection of the pesticide in different parts of the plant, examine the processes of growth, harvesting, preparation, or consumption to see if the pesticide is inadvertently or accidentally transferred from one part of the plant to another part or is altered in an unanticipated way, and measuring how long the pesticide remains before actually breaking down. Because of the role of society and culture in policy decisions, regulation has taken different paths worldwide. The US regulates GM products, whereas the European Union regulates the GM process. The FDA regards the key factors in reviewing safety to be the characteristics of the food and its intended use rather than the fact that the new methods have been used in its production. Therefore, U.S. GM food products are not subject to special regulatory approval or segregation if the constituents of the food are the same or similar to substances currently found in other foods. Nor are U.S. GM foods required to be labeled. In Europe, all GM foods are subject to special regulatory approval because they are made with the GM process, and they must be labeled as GM under the European Union Novel Foods Regulation. The difference between U.S. regulations and those of the European Union have created problems in trade. For example, problems arise in how to label U.S. food imported into Europe. This problem has been resolved for corn and soybeans by assuming that all U.S. corn and soybeans are derived from GM plants, unless they are certified as free of GM material or organic. Additional problems will likely arise as more GM crops are cultivated. So new challenges for government regulation are looming as biotechnology advances. Genomic data can match the right medicine to a specific individual. Genomic data can also use genetic markers to determine genetic predisposition to certain health conditions. However, if this data is widely available, it can also be used to influence the selection or hiring for certain jobs and the cost of health and life insurance for specific individuals. So, alas, we do not get away from this discussion of bioethics, and it will continue to be something that your generation becomes familiar with in many aspects, and not just creating GM products, but how this information can be used not only for the animals and the plants that are created, but for us as well. So think about